Welcome everyone to this last discussion on for this, and I guess the end of the meeting practically, although Nina will be coming in at the end to give us some closing remarks and reminders. So I'm going to turn my screen on right now and introduce the speakers and the topic for this panel. Yeah. Oh dear. I don't know how to move this. There we go. Okay, sorry. Yeah, so we have a panel discussion here on now implementing policies to support fair data. And what I've done here to arrange this is to invite 4 people who will be giving us perspectives from the on implementation and how it might happen from the journal perspective, from this perspective of an international scientific society, from a funding agency, and from industry. And I'm hoping they will. I've worked out questions with each one of them. So what I'll be doing here then is having each of them speak, but I'll show their questions on this slide so you can see them too. And yeah, sorry, I'm just a little nervous today. Okay, and so each participant will then answer their questions, and then at the end we'll have questions from the audience. So write up your Q&A as things are going along. And you could do that, or just raise your hand at the end, and we can answer your questions that way, I do believe. Okay, so the first person up is Sarah Callahan. And so Sarah, could you turn your video on, and I'll turn mine off, and everyone else can turn theirs off. And so this is Sarah Callahan, the editor-in-chief of a new journal from Cell Press called Patterns. Thanks a lot, Sarah, for being here. I'm trying to figure out how I can turn myself off. Okay, there we go.
Thank you very much for inviting me. It's lovely to talk to everybody in a virtual sense. So I'll start off by just saying what Patterns is. So Patterns, as Jamie said, is a new journal of data science that was launched earlier this year by Cell Press. We had our first issue go live in April, and it's a gold open access journal, so everybody can go and read it. And what I want to do with Patterns is share data science solutions across domain boundaries, because I'm sure I'm not telling anyone anything new when I say that everybody is dealing with so much more data now, and it's so much more complicated. But everybody's going around and they're dealing with the same problems in slightly different ways. So we're almost reinventing wheels, but because the wheels are different colors and slightly different shapes, they're not actually interoperable. But we can save an awful lot of time and effort and energy by sharing the data science solutions that have been developed across domain boundaries and learning from each other, and that will help with our standardization and stuff like that. Because fundamentally, data are the foundation of science, and without good data, we can't do good science. That's all there is to it. Right. So question number 2 is about data and code accessibility statements, which are required by journals. And it's a case of, well, why are they needed anyway when you get right down to it? So fundamentally, writing a journal article is a way of communicating your results to your peer group and the wider community. And it used to be the case, historically speaking, that you could include your data actually as tables or as graphs inside the content of your article. That's just not possible anymore. Data sets are too big, code is too complicated. It just doesn't work like that. But to ensure the validity and the verifiability of the scientific record, and to keep that scientific record maintained properly, we do need to be sure that the code and the data that has been used to provide the results and conclusions in a given paper is still accessible and still can be found 5 years from now, 10 years from now, 20 years from now, when an issue comes up with it, for example. So the data and code accessibility statement says in clear black and white in a very clear portion of the paper, this is how you can find the data and the code, and this is how you can get access to it. And that's really helpful for the reader, knowing how they can do this. And it helps with, as I said, verifiability and reproducibility in the future as well. So I feel like I'm going very fast, but let's not worry about that. So the next question for me was, what can journals do to ensure compliance with data standards? So this is a tricky one because basically the scientific article comes at the end of the research process. And oftentimes, if you're only starting to think about your data management and your data archiving policies and things like that at the end of the day of the project, then you're going to have an awful lot of work to do to go back and retrofit things and make sure everything is documented properly. But it's at this point that the journal editor actually has the authority and the carrot to wave in front of somebody. I'm not too sure whether it's a carrot or it's actually a bit of a stick, but it's it's the sort of thing where maybe it's a sufficiently large carrot that can be used as a stick. I'm not sure. Anyway, but the journal editor can say, oh yeah, just before you publish your paper, the final thing that you need to do is you need to make sure your data is fair, and we can encourage people by doing that. And kind of helping them to do that as part of verifying the scientific record, and ensuring the validity of it. Oops, have I gone past my time?
No.
Okay, cool. So we can mandate or strongly encourage compliance with FAIR, and we can push that as much as possible and go, well, ideally we want your paper to be published, but we also want your data to be fair, and your paper would be so much stronger if your data was fair, that sort of thing. But fundamentally, we are working with community standards, and we're part of the conversation as to what those community standards should be, because we want to publish the best and the most useful research we can. But we have to work with the community. And that ties in nicely with the last question on my slide, which is what's the process journals use to develop and require specific data criteria? And it's basically, we consult with the community, both with scientific researchers who are using standards, and also other stakeholders like research funders, repository managers, librarians, and all those sorts of people. And we also want to, and we do partner with other publishing groups so we can standardize and work towards best practice across the whole academic publishing community. So we want to encourage, it's in everybody's benefits to make communicating science better and more clear, and make the results of scientific research more valuable, more reusable, fairer all the time. So, so journals, we're working with everybody we can possibly think of, all the stakeholders we can think of, to determine what best practices should be for a given scientific community or research community, and help authors and researchers implement these best practices. Right, that's my questions. Thank you.
Thank you so much, Sarah. That was great. And I'm going to now ask our next panelist to turn on his video, and others to turn off their video. Jake, I don't know if you can hear me, but if you could turn your video off, that would be great. Yeah, so there we go. Yes, so our next panelist is Dr. Luis Eduardo Coelho Andrade, who's a professor at Universidade Federal de Sao Paulo in Brazil, and he is also the chair of the Quality Assessment and Standardization Committee of the International Union of Immunology Societies. So if you belong to an immunology society anywhere, you're probably also a member of the IUIS. Yes, so Dr. Andrade Professor Andrade will be telling us a bit about IUIS and answering some of these questions here. Thank you.
Thank you, Jamie. Do you guys hear me?
Yes, we can.
You can? Okay. So good morning, good afternoon, and good night to everyone. I thank Jamie Scott for the kind invitation, and let's go for the questions. So what is the IUIS and its overall mission? The International Union of Immunology Societies is an umbrella organization integrating regional and national societies of immunology throughout the world. It was founded in 1969, and it embraces 4 regional immunology federations: the European, Asia and Oceania, African, Latin American, as well as Canadian and USA societies, and 88 countries and over 60,000 immunologists worldwide. The missions of IUIS consist of 7 items: to promote immunology, to unite the global immunological community, create a global network of expertise in research, empower members to make significant differences, identify and address inequalities, raise awareness of interesting issues, and contribute to global health. The objective actions are to encourage cooperation in the field of immunology through our website, membership services, communication network, grants, awards, to organize international education in immunology through congresses, regional conferences, and topical workshops, to contribute to the advancement of immunological science through our journal, white papers, external relations, and public awareness campaigns. We work under a governing council whose members are elected and rotate every 3 years, and the modus faciendi operates through 9 independent committees, such as the Clinical Immunology Committee, Education, Publication Committee, Nomenclature Committee, the Quality and Assessment and Standardization Committee, Veterinary Immunology, Vaccine, and so on. IUIS has external relationships with the WHO and with the International Science Council. Frontiers in Immunology is the official IUIS journal. We also have a very dynamic and informative website, which I recommend you to browse, a quarterly newsletter, and the educational website Immunopedia. Like Wikipedia, we have the Immunopedia. Examples of recent initiatives are recent webinar series on COVID-19. By coincidence or not, there were 19 webinars. The Frontiers Coronavirus Knowledge Hub, training courses in several developing countries, the International Congress of Immunology in Beijing last year. That's the largest immunology congress in the world. This is a triennial congress, and of course, regional congresses. Now, for question number 2, what are the objectives of the Quality Assessment and Standardization Committee and the Nomenclature Committee? The QAS Committee's mission is to promote and improve quality assessment and standardization in the various areas of immunology across the world. The Nomenclature Committee has the mission of establishing a universal and consistent nomenclature for both molecules and cells of the immune system. These 2 committees operate through subcommittees. For example, QAS committee has 5 subcommittees: the autoantibodies subcommittee, allergy, complement, leukocyte, that is the CD molecules, and the big data in immunology subcommittee, our most recent one. The nomenclature committee operates through 10 subcommittees dedicated to specific areas. The QAS subcommittees establish and distribute reference biological material for standardization of immunoassays in each area. They also keep updated websites with consensus classification algorithms, recommendations, clinical associations, and so on. Similarly, the nomenclature subcommittees maintain websites and databases with structured information related to specific areas. On the third question, what roles can societies play in achieving fair principles for big data using immunology? Societies should play a crucial role in advocating, stimulating, and supporting initiatives to implement and publicize fair principles in all areas. As an example, much of the activities of IUIS committees and subcommittees address several aspects that directly contribute to fair practice in immunology, such as nomenclature, education, standardization, quality assessment, and publication. Big data and their associated metadata should benefit strongly from fair practice because diversification and heterogeneity in terms and units of measure severely hampers the widespread use and integration of big databases. IUIS recognizes the crucial importance of standardization in big data, and recently we invited Jamie Scott and Felix Brennen to put forth the subcommittee on big data in immunology as part of the Quality Assessment and Standardization Committee. And I am very happy to have you guys aboard. And by watching all the talks just before, I can realize that IUIS and FAIR principles have all to do with each other. Yeah. So that are my answers.
Thank you very much, Luis. That was great. Yes, so let me move on to the next panelist, Joe Breen. So Luis, if you could turn off your video, and Joe, if you could turn yours on.
Can you hear me, Jamie?
Oh, you don't have video. Okay. Yes, so audio, turn on your audio, and please go ahead. Joe is the, he is the section chief for immunoregulation in the basic immunology branch of a DAIT, so that division of allergy and immunology and transplantation at the NIAID of NIH. Joe, thanks for being here.
Jamie, can you hear me now?
Yeah, we can hear you just great.
Okay.
And we can
Thanks.
see you.
Oh, great.
Your handsome face.
Oh, that's very nice. Well, thanks, Jamie, for the invitation and the other organizers to participate today and actually listen to the previous days. My name is Joe Breen, as Jamie mentioned, and I work in the basic immunology branch in what's extramural NIAID. So we're a funding portion of NIH, and we support majority of the basic immunology grants that are funded by NIAID, split among a few colleagues of myself, about a dozen actually. And the reason I'm here to talk to you today is that actually I am the program person responsible for the IEDB. Although I didn't conceive and start it, I took over that function about 5 years ago. So I worked very closely with Bjorn Peters on that and some of the other efforts that he and Patrick talked about earlier. My division actually supports import, which you've also heard about. So I want to talk to you a little bit about some of these questions that Jamie prepared ahead of time. And I want to remind you that I work in the Division of Allergy, Immunology, Transplantation, and I can give you my perspective. But I can't really speak on behalf of all of NIAID, but I'll try and point you towards information where you can see the relevant strategic plans and other publicly available documents to give you the resources where you could do that. So what is DATE's approach to building collaborative work and data sharing among COVID-19 basic science and or clinical researchers? So we actually early on in the COVID-19 pandemic, there was a blog post posted by another NIAID colleague I'll talk more about in the Office of Data Sciences, encouraging collaborative work and data sharing and listing quite a large list of repositories that are data specific, such as import for immunological data, IDB for immune epitope data, but also a whole host of others across the NIH, where folks were encouraged to put their data and definitely in response to COVID-19 as well as normal. Although, to be honest, at that time, mostly the only work that was going on was COVID-19. Many other places had shut down. But since that time, I think we have seen quite a bit of data sharing, and it was referenced in an earlier talk that many journals are making publications, and then the pre-publication servers as well are making COVID-19 data. So I think it is in the spirit of collaborative and data sharing, at least as far as figures go. And there's a new resource called the National COVID Cohort Collaborator, or N3C, that's being run out of, it's not a NIAID program, it's run by another institute, NCATS, that is publicly available and is actually for clinical research and encourages collaboration and really making connections, and is actually building a very large database as well. I would encourage anybody to look there. So what are the current requirements for data sharing in NIH standard R01 grants? Well, that is actually under revisement. Currently, if your grant is less than $500,000 in direct costs, you need to have a data sharing plan in your grant. It's not scored, but it's required to have it in the application, and then the reviewers make comments. If it's larger than actually a larger budget size, then actually there are more requirements. And the other piece here is that for programs where we specifically ask for applications, which tend to be either R01s or cooperative agreements, we, NIH program staff, define what data sharing requirements are up front. And in these programs, we require data to be deposited into import in most cases. And the language actually says into repositories, such import or other publicly available repositories as appropriate. So we're not duplicating things that should go into dbGaP, for example, or GEO, or other places where there are already substantial investments. But in this way, we actually do require that for large programs and actually for contracts as well. So number 2 is building infrastructure for big data, a goal within DAIT. And I think that's true. I mean, basically the programs I mentioned, import and the IDB, are 2 examples of that, which are actually fairly long-standing now. But more recently, there is an Office of Data Sciences, and actually I discussed these questions with a representative from that office, Ishwar Chandra Mulaswaran. I had to look at that because it's a big name. Who is there? Just the fact that NIAID has this office, I think, is an answer to the question of whether this is a goal for NIAID, and really to develop over time a data commons approach for all of NIAID. And DATE would certainly be part of that, and the things that we've built already, such as import and the IDB, would definitely be part, and hopefully moving forward be all interoperable and findable, and really increase the utility for all those in the immunology side, but also all the metadata, and hopefully connected at some point to an NIH data commons. So the third one is NCBI's Sequence Read Archive is an important resource for researchers, but its data sets are inconsistently curated, making them hard to work with. What are your thoughts on improving sequence data consistency and shareability within DATE? Well, certainly we encourage, I guess the short answer is that for the sponsored programs that we outline what we want people to do, we certainly want them to follow the consensus that's been developed by members of AIRR and others in the community, and the consensus document, so that there is commonality for curation and metadata to increase the value. And I think the more that people do that, then obviously that strengthens the ties between databases. And I think Felix mentioned earlier that, you know, right now it's a federated idea. It crosses several different databases, even the outside of NCBI. And I think we have to continue to push for consensus. That's why I participate in this, and I think it's important. And I think we need to get there because obviously the data is growing quite quickly now. And I think there's been some reference to we need to be prepared for the next pandemic. So we need to keep moving down this pathway. So I think I'll finish there, just say that I think these things are important. They're certainly recognized important to date and certainly to NIAID based on the resources that we've put into it and hope to continue to. And I have a few links that I'll put in the chat relative to recent notices and the blog post I mentioned earlier relative to number 1. Thank you.
Well, that's great, Joe. Thank you so much. And I forgot to mention in introducing you that Joe was there from the beginning for the AIRR community, and John Warren as well. They made sure that we got funding, well, not funding, but a place to hold 2 of our original meetings. And that's been really special. But also, Joe was there for our very first meeting, as a matter of fact. And when everything was all over the place, and we originally, you know, spent 3 days, you know, arguing things and going, what are we going to do? What are we going to do? And then it all just kind of came together in the last day, as I recall. Anyway, so thank you very much, Joe, for participating.
Hope we can have a meeting like that again soon.
Yeah, I'm sure. Well, we're definitely going to have a virtual one. That would be a lot more fun to do it face-to-face, though.
Yeah, I was mentioning the face-to-face, but
yeah,
virtual's not bad. Thank you for the opportunity. Yeah.
Yeah, thanks. So great. So let me just move on to our, sorry, there we go. Our last, but not least, speaker. So Joe, if you could turn yours off, and Jake Glenville. So Jake is also a long-term in person supporting the AIRR community, but also a pretty amazing bioinformaticist and immunologist in his own right. And he's also the founder of 2 companies and the CEO that are coming up with antibodies, new therapeutic antibodies and vaccines that are pretty amazing. So Jake, please go ahead and introduce yourself and so forth. Thank
Great.
you.
Thanks for having me on, and thanks everyone for your time. So yeah, I'm Jacob Glenville. I've been involved in repertoire engineering since kind of early on in 2008 when I was developing methods on 454 sequencing instruments. And ever since, I've always tried to figure out what were the coolest ways that we could use this new class of information to ask aspirational questions like the nature of diversity dynamics, but also very practical, roll up your sleeves questions like, can we make a COVID antibody to treat the world faster? So my company's Distributed Bio and its therapeutic spin-out Cenovax, they're a continuation of that work. We apply computational immunoengineering. If you go into our laboratory, you see we integrate high throughput sequencing at every stage of characterizing large numbers of human repertoires to understand diversity dynamics. We use that information to then build physical phage display libraries that have better combinatorial distributions of diversity and therapeutic strategies, and we check our work with deep sequencing. We then deep sequence the outputs of panning selections on robotic bead manipulation devices that let us run 24 different pannings in parallel. But the critical thing is we then integrate functional data into those clones. So I think we've done full kinetic characterization of over 50,000 clones. We have a panel of a whole bunch of kinetics devices, FACS screening instruments, expression characterization. To the extent possible, I try to integrate high throughput data with the repertoire sequencing and output functional characteristics that we can associate. And because of that, the consequence is that we've been able to run with a pretty small team over 50 programs, different targets in the last 12 months, including really challenging targets like GPCRs and peptide MHC complexes. And that's really a consequence of being able to use repertoire sequencing to look deep in repertoires, figure out where they were broken, and find ways to improve those libraries using math, and then, essentially it's PCR overlap extension with good designs of the components that you add together. So that's the history. The specific thing you may have heard about us this year is that we took this technology platform and in January, we applied it to going after the novel coronavirus. And the cool thing we did that other groups were not able to do was we took these, as a consequence of, again, repertoire sequencing, a bounty of the work that you all do, is that we used anti-SARS antibodies from nearly 20 years ago from the SARS outbreak, and we were able to strategically design libraries of billions of variants, which were able to search deeply and span the evolutionary quantum leap from SARS recognition to binding and vigorously neutralizing the novel coronavirus. Those were developed very quickly and optimized using the same process. You build billions of versions, we can span to go bind the new variants. So we're optimizing, and then we use deep sequencing to further look before and after the selection pressure to teach us what were the positions that were under selection, what were the positions under neutral drift, and this gives us this powerful ability to optimize those clones further without having to do iterations, which would have been not reasonable during a crisis. In fact, Dr. Jean-Philippe Becourt got a special Belgium embassy national interest exception to come back to the United States. And he's got one of these Einstein 01 visas because he's leading this work in our laboratory and being able to use deep sequencing for the very practical but critical application of doing a single fast evolution and optimization of a lead. So that's sort of what we do at the company. We were able to complete that very quickly. So that was done in March. We had our leads that were cross-optimized towards the novel target, and we sent that out to a collaborative network of 2 national labs, 2 universities, and then a company, and actually 2 companies, so 6 groups in 3 nations to validate the work. And that's part of our strategy, again, is that we feel that it's better to have independent groups because everybody makes mistakes, so you can kind of get the boosting benefit of different independent groups validating that we neutralize and we provide potent protection. So that's who we are, and I think we just speaks to the blue collar value of like there is direct actionable information available in these repertoires, and it's speeding up our ability to make medicines faster and engineer them to new purpose. And the really cool thing about what we're doing is that we can apply that in future pandemics. So I guess that gets us to the second question. Your business model aims at developing low-cost immunotherapeutics and vaccines for use worldwide, including in low to middle-income countries. What is the importance of technologies like high-throughput sequencing and computational systems approaches to the cost of developing new therapies and vaccines? Please provide examples. So my parents are Americans, but they're hippie expats. So I grew up in Guatemala in a Mayan village, and that has left with me the impression that medicines, in order to be useful, shouldn't cost so much. We're the underdog with sort of a bone to pick with respect to the COVID therapeutics, and specifically my big problem with the therapeutics is that you see with remdesivir, they're charging $3,120 per dose. I'm afraid the same thing's going to happen with the antibody therapeutics, where typically they charge $5,000 to $8,000 per dose. That is ridiculous. It costs $100 per dose to generate the cost of goods for making an antibody. The rest of that price is really the calculation of the amount of pain an insurance company is willing to put up with in the United States for those lucky enough to be insured. We need to rethink that. And we're working on doing that. I would say a testament, again, to the work you all do is that we were just as fast as other companies that had $25 million upfront thing. I'll just call this P3s, this pandemic preparedness. They had pandemic preparedness money and we did not. And yet we were just as fast as them as producing molecules without that money. So that's the power of repertoire engineering. It's $25 million of savings on a group of 35 people, and we had other programs. We're working on like 40 other discovery programs for Pfizer, Boehringer Ingelheim, Gilead, Teva, other companies at the same time. And that's where I think if you really give a head-to-head of these kinds of technologies, this is where the future lies, because having access to large data and rapid screening and single-pass optimization is really bringing therapeutic discovery to the digital age. And if I had even a fraction of $25 million, I would have beat those other groups by far. Now they are beating me now in terms of getting to clinic because they got another 400 million. So the benefit only goes so far, but I will put to you that this is the LED rather than the old school light bulb. Now, so the way we do this is we use data at every step, like I mentioned. We use data to help us understand the diversity of the repertoires. These are simple things like how many affinity-metric variants are there in CDRH1 of VH3-23, and what is the distribution of mutations, so a directed acyclic graph away from germline, how many germline variant alleles exist in populations, and what is the proportional distribution, a percentage of the repertoire within a given person and across a population that say is germline. And so you guys say 25% of CDRH1 is germline, another 15% is distributed across single mutant variants. Once you know that information, you can then simulate what a library would look like if you adopted it. And you can also ask the question, well, what if I doped in synthetic extra copies of certain tiers of mutation or germline residues, or removed some, and I built a library that was better, or I could shift the distribution to solve certain engineering goals, like spanning the evolutionary leap to recognize the novel coronavirus. I think in terms of cost of goods, that saves a huge amount of time because we're able to do more with less people and less time. That means you don't need 30,000 people to go run these projects. You can do them smaller and tighter. That means that you don't have to charge so much for the medicine, which means more people can use it. Beyond the work I do personally, there's other areas in this field where synthetic biology is really helping accelerate and reduce the cost of drugs, which I think ultimately is going to be something critical towards making a healthier humanity. I also think it's good business. If I can make more doses and charge less, I think that's something we want because it gets the world back to work better and faster. We can have that in-person meeting again. And people are going to be more likely to take the medicine that works better and it costs less. Now, it doesn't going to solve everything. So computation right now, it's a fantasy to think that there's a magical program where you can just put in an antigen sequence and it'll spit back the sequence of an antibody that recognizes it. But you can create a mixture of using computation and deep sequencing and integrated data to guide your discovery process. It's like playing poker with a computer in your pocket. You still have to sit at the table, but you have better odds of success if you have that computer next to you. So the last question is, from your perspective as CEO, what are the pros and cons for the biopharma industry of following FAIR principles with respect to high throughput sequencing data and AIR sequence data in particular? Please provide examples. So the obvious thing that I would say here would be industry wants to have new data, but they're afraid of sharing theirs. I think I actually want to flip this conversation, and I think you should be more aggressive and ask, what can industry do for you? So there are two kinds of data, the repertoires that are, I would say, of obvious blue collar, roll up your sleeves value. One of them is obviously specificity repertoires. So we've run 50 programs in the last 12 months. I think we've run over 80. Each one of those we've deep sequenced. So we essentially have 80 repertoires, each one deep sequenced, thousands of hits against a given target. And that is immensely valuable. Imagine that you had collections of hits that you could begin characterizing convergent specificity groups that recognize hemagglutinin or HIV or smallpox or chickenpox, whatever. And smallpox is an odd example. But you could begin then painting the repertoires with specificity based on convergence groups of detectable similarity. And with enough examples, you will win that war and you'll be able to assign function directly from the primary sequence. The problem, of course, is that companies are going to be resistant to releasing that information if it's about their precious targets. I would hope that that is less of a problem around pathogens, and that could be immensely valuable for, I say, oncological phenomenon pathogens. Even things like anti-double-stranded DNA binders, which pop up in lupus, would be a really interesting database to be able to mine. And that gets me to the second type of repertoire, which is the general property selected repertoire. So I think it would be very valuable for the whole community to have access to like thermostabilized repertoires or acid resistant repertoires, or generic things like maybe anti-double-stranded DNA or a disease where you don't know the exact target, but you know that certain clones are enriched. And I think those are the things that are the easiest for pharma to contribute. But I think that the AIR community should ask and argue that it is a collective goods benefit that industry contributes their like I can run a selected repertoire against double-stranded DNA, no problem. I can do it as a side project along with a bunch of other things I'm painting at the same time. And there's other companies that can do that same thing. And by then contributing that information to the AIR community, what they will get back in exchange is essentially a screening tool where they can look at their sequences and they can ask, okay, I should kick out certain sequences because they look polyspecific, they're generically hitting charged surfaces. This one showed up in this, it's a sequence activity relationship models of low stability proteins, for instance, low stability antibodies. So I think that would be the value proposition I think would make sense. And I think as long as you're not going after their precious targets, I think companies are going to be willing, they're going to go, the value of learning something about my antibodies early without having to do as many experiments, or at least triage early, will outweigh my risk of sharing something that isn't really that valuable to me anyway in isolation. But if we share our data, it would become very valuable. So that's my perspective on
Great. Thank you so much, Jake. That's really, really inspiring. Yes, so I'd like to now just ask all of the panelists to turn on their videos and their microphones. And I've got one question from, what do we have from Bjorn, that I think this is for us, right? Why are data sharing plans not scored? That could be a game changer.
Maybe I should take that
Yes.
since I said it.
Please do.
Okay, it's a great question. So it's for grants and contract applications where they're solicited. So we write an RFA, for example, for specific area or a contract for specific purpose, then we can require that to be part of the evaluation. Right now, that is not part of a grant that comes in that's smaller than 500,000, because that's the data policy that's been in place since 2003. But those are being currently revised. And actually there's draft proposals that are available for comment. So I would encourage anybody who's interested to do that. I don't know, honestly, I don't know where that's all going to fall, but obviously things have changed a lot in 2003. And I didn't mean to give the indication that's never the case for all the solicited programs and the contracts, things like the IDB, for example, and lots of other programs that we definitely would take that into account, and it would be part of the scoring. So I hope that answers the question.
Yeah, so that's a pretty good answer. And we just have a few minutes left, and I have a question for all of you and for the audience as well. And that is that in the back of my mind, because this is just kind of what we do in the meetings group, as we kind of think about what do we want people to talk about? Like, what kind of problems do we need to be thinking about? And that's where this whole panel meeting came from, this whole day of discussion. And what I would like to ask you is, would it make sense? Because, you know, everybody's in their own, I won't call it a box really, but in their own kind of area or field. And there's not a lot of necessarily interconnection between societies and journals. I mean, the funding agencies, industry, and I left out institutions as well, like, you know, universities, for instance. So what I'd like to ask you guys is, does it make sense for us to be thinking about holding a workshop on this kind of issue? Thank you, be holding a workshop on this kind of issue where it's, you know, broken down to more discrete questions, but the idea of trying to build more of these interactions so that we can get more kind of, I guess you'd say, international or group consensus, possibly, and feedback. For instance, I could see that there right now, for instance, at NIH, I'm just making this up as a possibility, there's not necessarily a very concerted effort for building the infrastructure we need for the next generation of bioinformaticists and so forth, to really be able to pull things together in ways that, for instance, the AIRR community has done. And we have gotten help, but it's been, you know, more just generous people than necessarily built into the infrastructure. So, so I'd like to see if any of you have any feedback about that general idea. And audience, raise your hand and participate. So the idea is perhaps putting together workshops in the future on these kinds of issues.
I'll jump in there if I might, and with the understanding that I am a bit of an interloper because I trained as a physicist originally, but I can say that yes, this is a very good thing to be doing as a first step. I've got a lot of experience working with around this exact topic in other domains, specifically earth and environmental sciences. And the way to start building consensus is to start talking about things, and the way to deal with the whole notion of fair and try to improve things for everybody is by getting people together and talking about them and coming up and with developing standards and coming up with a way. It's the way I always think that we want to make it easy for people to do the right thing, and that means talking to people with regards to what they currently do now and how we can nudge them in the right sort of direction to make best practice easier for them to actually achieve. Because you're not going to get very far if you're coming down and waving a big stick around and saying you must do it like this without actually talking to people. You need to get their buy-in. So absolutely, having a workshop is an excellent way to start.
Well, we're pretty much done here. We're a little over time, but does anybody have any other comment to make?
Just one comment, Jamie. Your idea of a workshop is very welcome by IUIS. So maybe,
At the
we
next
have a
Congress.
test together. Yeah, yeah. Very good. That's it.
Thank you, Luis. Jake.
Yeah, I think I would second that, especially given patterns. I think we're at a point right now with repertoire data and the community, the AIR community, where integrating data across disciplines is like this is the right time to do it. There's now in place, we understand the sequence activity relationships, and we have good command of sequence structure function instruments. And I think the community is very powered with computational analysis. So what is needed now is additional integration to additional data sources that can enrich our utility of these platforms. So one example would be go to people who have lots of, let's say, stability data. They have both the data, which we need, and then second, they have the pain of knowing how annoying it was to generate that data. And so therefore, then they would love to find computational guided tools, models that be able to predict, okay, these are the properties of stable and non-stable clones, which you can now propagate onto hundreds of millions of sequences without having to go generate that data piece by piece. And so that would be an example of the kind of gathering that one would envision on many different axes of information and functional characterization of these repertoires.
Great. Okay, well, and Joe, do you have anything to throw in there, or are you good?
I mean, I think the activities that you've been doing, you and others, Jamie, is exactly what we should keep doing, because I think that communication and harmonization starts with people agreeing on a direction, and I think that's what I'm seeing here. And that's why you mentioned John and I, and we've tried to support it by providing a place to save some costs on meetings. And hopefully we can do that again.
But it was also inspiring the support and direction that you guys have helped us with. Okay, well, so I need to move on now. Thank you, everyone, because we have to end this meeting.